people are in.
Okay. So now we have the recording in Zoom, and now you hear the cling.
Yes.
When the people are coming.
Yes.
Okay, maybe I should put mute all. Okay, mute all. Okay, let's see if the cling is still coming. Okay, and then I'll put now live in Facebook. And wait a second to see if it's going live stream on Facebook. Okay, so therapy against okay, go live. Okay, so it will be coming in on Facebook. So don't say any secrets, big secrets during the conference. If you have any secrets, don't tell it, because all the rest of the world will hear them. Okay, wait a second. Okay. Do you still hear the clink clink? Oh.
Yes.
I don't know where is the setting to
No, I think you should not leave us, let us as hosts, but just normal, but to give us the panelist.
Yeah. No, you are a co-host and also
Okay.
Otilia. It's like panelist. It's the same. The same.
And just give us permission for the for sharing the screen. Yes. Okay.
Yes, you have the permission to share the screen now. And I'm sorry for this clink clink, but I don't know how to stop the clink clink.
That will be all during the.
Okay, suppress background noise with the medium. Okay, ringtones. Join audio by computer and joining a meeting. Mute microphone when joining a meeting. Okay, so it should be now okay. Let's see.
We discover step by step the secrets of technology.
Yes.
Okay.
Yes, it should be okay. Let's see now, because I will admit 2 more people. Oh, you still hear the?
Yes.
Oh gosh. Okay. So unmute all. Okay. Now everybody is muted. Cristina, unmute. Please unmute yourself.
Yes.
And let's see another one. It's clink clink. Okay, wait a second. Okay, anyway. Oh, you still heard the clink?
Yes.
Let me try one more, and then if not...
it's like this. Yesterday was the same.
The same, okay. Audio, okay. Test microphone, automatically adjust, suppress background noise. Low, medium, high. Okay, I put here. Show meeting option. So, okay, more settings. Echo cancellation, aggressive. Okay, I'm really sorry about this, but let's be flexible.
Yes, we'll try to manage, don't worry.
Yes, it's like a good sign. More people are coming. We should be happy.
Yes,
okay, so please, Cristina, start your presentation.
First of all, many thanks for letting me participate into this event, which is already a success year by year. Unfortunately, we could not meet each other, but we hope for the better. Who knows? I will share my presentation. Wow, not screen, but what about? Just a moment. Files. Microsoft. I don't know why is this happening. One moment, please. Today it's after the eclipse, probably. Okay, let me see if I can join now. Okay, just I will try to share now. Is it okay?
It's coming. Yes, okay. Now it's perfect.
Okay. So this is a presentation. It's just a review of the main techniques and technologies that are now used in the pharmaceutical industry in order to try to enhance the bioavailability of some of the most important pharmacological active compounds in bee products. And we will see that this is in full progress to get the better way to penetrate our body and to make the best profit out of it. We have seen that many people are buying, let's say, pastry or other things that are powdered with the so-called honey, powdered honey. What is dry or dehydrated honey? So I wouldn't call it anymore honey, and you will see why. Because after the spray drying process, when we get a fine powder from the honey, we use several stabilizers like weight or starch, or maltodextrin and fructose, just to make a 1-to-1 ratio to have like a powder like table sugar. But this, it's indeed benefiting because it is not sticky, it's mass-free, does not see any crystallization issues, and it's easy to clean up. But should we call this product honey anymore? It's just a question, and I would like to see your feedback. Then honey is known that used in natural form, it can be also stabilized with some other agents to become an excellent wound healing agent and a good antibacterial. There are some products that are already now, and my last, I say among the last papers that I have published was related to the defining the grade of medical honey. And it was together with a company from Holland who worked in producing such plasters with honey. Well, honey can be used also for diagnosis using a special technology, the nanotechnology. And it is mostly important for the diagnosis of cancerous disease and the progression of the disease, because we know that imaging sentinel lymph nodes provide the best information about this progression of the disease. What is very interesting is that it's a synthetic methodology that has been created to develop luminescent carbon nanoparticles from food-grade honey, so normal honey, which offer a rapid clearance and a high real-time, high-resolution photoacoustic imaging. This is a good hope for the future to have a faster diagnosis of the disease. In cosmetics, we know that also phytocompounds are loaded on nanoliposomes. The same can happen with honey, and the results are very good. It's easier penetrations because liposomes have the same structures as our skin. Bee pollen. the main directions of the research for bee pollen are towards development of new technologies for pollen outer casing disruption and removal of this part of the pollen external wall. Technology for enriching the pollen extracts with such active compounds or other active compounds. Then there are studies on the biochemical parameters compared with raw pollen with the pollen extract. Technologies of oil extraction of bee pollen, which seems to be also very important in several therapeutic applications. Then it's the development of technology for wasteless processing of bee pollen and development. I can tell you that in my activity, as I already mentioned yesterday, we used the last from the pollen extractions for preparing some food supplements very rich in amino acids, proteins, and minerals. There are also significant progresses made in the development of the technology for the exine disruption. And one of the methods that will be specified is the fermentation with different agents. So if we obtain such a pollen extract in which the outer wall is disrupted, we can have a high stability, a preservation of all useful properties. We have a low allergenic activity as most of the allergens in the outer layer in the sporopollenin are gone. We have a high concentration of valuable nutritional components, high digestibility, high level of sterility, like the same after fermentation, like with bee bread, which is obtained inside the hive. Using the hydroalcoholic extraction, we have up to 96% of the exine disrupted. Bee bread extraction technology. We know very well that it's very difficult to take out the bee bread from the cells of the comb. And that is why there have been many minds put in action to try to get an easier method to take it out. One of these methods is proposed by Akhmetova and co-workers. It was published in 2012. The technologies consist in soaking in water the bee bread combs. Then we can make the manual extraction through vibration, I mean, just vibrating. Then drying and separation the granules through vacuum. Again, drying, and then freezing, sedimentation, and filtering of the wax particles. The obtained preparation is put to acoustic drying, in which 25 frequency ultrasounds are used. Using this acoustic drying procedure, the humid air is out of the product and also the particles of beeswax. Finally, what we get, it's a enough pure concentration with more than 85% of the bee bread granules, which are entire and not cracked. Propolis is also used with the nanotechnology, and there are several also propolis extraction methods like ultrasound technologies or supercritical carbon dioxide technology. All technologies are meant to reduce the extraction time and to increase the extraction percentage. With the ultrasound technology, we can use only 15-60 minutes of extractions, while with the classical extraction, the time is longer. After applying the technology, of course, the extract was checked for its antibacterial and antifungal activities, as well as for the antioxidant activity. This is a scheme for the preparation of nanoparticles from raw propolis. There are several steps like extraction with ethanol and reprecipitation to get propolis particles. Then these particles are resuspended, that's supposed to a freeze drying process, and then in nano suspensions. With the nanoparticles, cytotoxicity of propolis nanoparticles were checked also against several cancer cell lines. Then the activity, antimicrobial activity, all the results have shown that these nanoparticles are really more active than the original extracts. Another technology is to prepare propolis liposomes, which also used by ethanol injection method, together with lecithin, cholesterol, and propolis flavonoids, we can obtain spontaneously after further evaporation of the residual ethanol, the liposomes. The resulting mixture is homogenized with ultrasonication for 30 minutes to form the small single chamber liposome. There are different sizes of the particles from 0.22 up to 0.8 microgram. Membrane filters, millipore, that was used to filter the extract. There are different such encapsulations of propolis liposomes, which encapsulation is a critical factor to appraise the quantity of order to achieve high encapsulation efficiency in the experiment preparation conditions. So in vitro, the propolis flavonoid liposome not only could significantly promote T lymphocytes proliferation singly or synergistically with phytohemagglutinin, but also could increase the expression levels of interleukin-2 and IFN gamma messenger RNA, demonstrated the stronger immune enhancement activity, which provides the theoretical basis for the further experiments in vivo. Propolis solid lipid nanoparticles are proposed as bioactive medications for topical intranasal therapy. We see that there are many studies now to prepare even a vaccine with such an administration, a nasal administration. So the studies that have been carried out in 2015 showed that propolis as a lipid bioactive nanocarrier can be used when drug delivery. Several active compounds like diclofenac and also the flavonoids can be used for the local treatment of nasal cavity diseases. Then the polymeric nanoparticles of Brazilian red propolis is another papers that have been published. The polymeric nanoparticles loaded with red propolis were prepared with a combination of polyepsilon caprolactone and fluoronic with nanoprecipitation method. The flavonoids were included into a polymeric matrix, and several flavonoids have been identified in the ethanolic extract of red propolis. Sorry. What is fluoronic is a poloxamer or amphiphilic copolymer composed of a central hydrophobic chain of polyoxypropylene flanked by two hydrophilic chains of polyoxyethylene or polyethylene oxide. Propolis hydrogels are also used in wound care dressings containing propolis. There was a study that have been published in 2016 in the UK, in which such gels were loaded with propolis and used in several treatments, especially for wound caring. Of course, there are also gingival gels with nano-antioxidative gel. The technique was nano-biofusion technology. This is a result of mixing two top technologies, and the product contains antioxidants in nano-emulsions. Of course, these are the antioxidants specific to propolis. Also, this propolis can be used together with gold or other active compounds for cosmetics. So masks with hydrogels with calming and soothing effect on the skins can be prepared. For corneal microbial keratitis, there have been some nanofiber prepared to repair such disease, wounds in such diseases. We have the experience of using water-soluble propolis in treating keratitis, especially herpetic keratitis. Probably Stefan remembers the way when we used also the nebulizations with hydroalcoholic propolis. The propolis was mixed with gelatin and polyvinyl alcohol to produce biocompatible nanofiber patches fabricated via electrospinning technique. As you can see here, there are several fibers. The gel patch can be an ideal matrix for 3 and up to 5% propolis addition. The studies have shown that at 3% concentration of propolis reinforce the mesenchymal stem cell proliferation and the humidity, wettability properties of the patches, so they still should be wet. Royal jelly is used and was used for adhesive films, especially in the treatment of oral mucositis following the treatment of cancer, head and neck cancer. That was published by Watanabe in 2013. From then, this has been more and more developed. Some techniques of extracting royal jelly, as many specialists says that the contact with air of the royal jelly can damage the concentration, can affect through oxidation the properties of the active compounds in royal jelly. So there have been some technologies developed to exclude the contact of royal jelly with air oxygen. One of the methods was using noble gas like argon. And another one, which is in Russia, I still have to look for it, enables to exclude the contact with air oxygen of the royal jelly. I did not see the description of the technologies, but I have mentioned it as it is published on a Progalski site. In royal jelly, there have been several proteomic analyses that reveals novel proteins and potential phosphoglycoproteins, as said in 2008 for Asavo. From then, many other studies have been carried out showing that there are royal jelly major proteins, especially major royal jelly protein 5, which is a reliable freshness marker, and that's the best way to maintain quality of royal jelly under freezing conditions. If we check this parameter, we see if the royal jelly is fresh still after freeze drying. Also, royal jelly can be used in several cosmetic applications. It can act as an antibiotic, and we know that 10-hydroxydecenoic acid, the active, the most important fatty acid in royal jelly, offers such important activities. When used on skin, royal jelly works to promote cell and tissue repair, which is, while in the same time is stimulating the collagen productions. It seems that both vitamin C and 10-hydroxydecenoic are responsible for increasing collagen productions. We know that royal jelly is very useful in many cosmetic preparations, an essential aspect for fighting especially wrinkles and tired age skin. In the liposome technology, the so-called nanoliposome technology, the 10-hydroxydecenoic acid can be encapsulated. In this way, we can use it better with a specific target. The composition of the synthetic material to which 10-hydroxydecenoic acid is added is butylene glycol, sebacic acid, and 1,10-decandiol. There are other liposome technology. We know why, because we spoke about liposome as similar in the composition with the lipid layers like the skin. So it's a royal jelly lift concentrate, which works against wrinkles. I have seen many such results in Korea, in Japan, especially South Korea. Now, the Be Venom, the nanoparticles of Be Venom. We know that there have been made many studies showing that such nanoparticles loaded with B venom, but not exactly B venom, melittin, can kill the human deficiency virus, immune deficiency virus. But this is only, it's a targeted product because it does not affect the surrounding cells. It's an important step toward developing also a vaginal gel that may prevent the spread of HIV, the virus that we all know causes AIDS. The action of melittin is directed to the double layer membranes which are attacked. And this concept is not limited only to HIV, because many other viruses like hepatitis B and hepatitis C rely on the same kind of protected envelope and would be vulnerable to melittin-loaded nanoparticles. This is just a scheme to show the melittin in liposomes and also the melittin in nanoparticles. It is just a short presentation to see that the pharmaceutical industry is developing, that our products are taken into account. So this is a hope for, let's say, to have some pharmaceutical products with B products. It gives the opportunity for new formulations and the diversification of the medicines based on natural products like the very valuable B products. I want to thank you very much for your attention.
Yes. It was again a wonderful presentation. Mrs. Cristina Mateescu presented the same thing yesterday in the Romanian language. We Romanians are so happy, so how do you say this? So lucky to have people with a lot of knowledge on beekeeping, and Mrs. Dr. Mateescu has a huge knowledge on bee products. And you just saw in the presentation, very clear, very interesting, a lot of new technologies. For me, it were many Chinese words, the presentation. So it's a top, very modern. And I'm sure in the future, this kind of products will be a lot used in medicine, hopefully also in the hospitals, in the surgery, and so on and so on.
Let's hope it's a gate for our products to penetrate into the pharmaceutical industries, because otherwise we will still remain as food and food supplements with the bee products, unfortunately. Yes. Not with the actual legislations and regulations.
Yes. Any questions from the participants now to Cristina? Or we go, oh yes, one question from Frederick. Frederick, I believe you are from Kenya, yes? Can you activate your microphone and your camera? Yes, activate your camera and also open your microphone. Oh yes, now we can see you. And yes, okay.
Hello. Yes, good morning from Kenya.
Good morning.
Yeah, it is afternoon now. Now, thank you very much, Madam Cristina, for the good presentation. And I am very much interested in this propolis extraction. Now, I don't know how we can get some procedures on how to extract both liquid propolis and also powdered propolis. The second question is about which method is most effective between alcohol extract and water extract. Thank you.
Okay. Well, along the time, there have been many technologies proposed for propolis extractions. First of all, it was the 96% alcohol extraction. I tell you from my experience that when we approved Propolis tincture as a medicine in Romania, that was some times ago, we were forced to use 96 alcohol and not 70% alcohol as is proposed. And I'll tell you why. Because the people in the National Institute for Medicines said that 70% alcohol has its own antimicrobial activity. So when using together with propolis, they could not make the difference between the propolis tincture and the 70% alcohol. So as a result, they said, show us the results and the antimicrobial effect of propolis using 96 degrees ethanol. So that was the procedure in that moment. Of course, we know that 70% alcohol, which contains also water, does permit the extraction of both some hydrosoluble compounds, because there are also phenols, polyphenols, flavonoids that can be extracted into the water phase, and also flavonoids that can be extracted in the ethanolic, organic solvents. To get it, as you said, in powder, that can be used after a very good extraction. We made it with special solvents, but passing also through the ethanolic extractions. And then we have lyophilized or freeze-dried the extract. What we can get is a spongeous powder that can be easily dissolved into physiological saline or distilled water to be used in several treatments that were used for nebulizations, because we know that propolis is sticky, and it helped us a lot in treatments of eyes diseases as well in nose diseases. In otorhinolaryngology, we use a lot these products with the lyophilized propolis extract. For the extraction technology, you can get in touch with me. You find my my email on, or I will write it on the chat. So anytime you have any questions, please address freely. Thank you for your questions from Kenya. I hope I have covered a little bit of your interest. Okay. And Stefan, you are muted. Stefan, you are muted.
Yes, yes. Yes, yeah.
Okay, sorry. Okay. Stefan, maybe I want to ask to Dr. Christine Mateescu. Can you
Yes,
help
please.
me?
Yes, we can hear you.
Okay, thank you. Thank you, Dr. Christine. I'm James Hutagalung from Indonesia.
Maybe I'm sorry I could not join your symposium. I'm really very sorry.
It's never mind. I like it to ask you. I have 2 hospitals in Indonesia, Surabaya. The first from the university, and the first in the government in area beside me. So I like it. More people in Indonesia more sick about HIV and COVID. So do you like it? Your collaboration with Tropical Disease Center with maybe Dr. Stephen and you. I like to invitation to you and collaboration, because I'm more in Indonesian more people maybe. Do you like it? So
yes, it's very interesting. Thank you. I would like you, Professor Hutagalung, to be also a correspondent member of our commission of Apimondia. That will be very helpful to know how apitherapy is developing all over the world. And I want to really promote people from all over the world to work together with us, exactly as in International Federation of Apitherapy. There is no competition. We can work together to promote apitherapy. And
I like it. No competition. So I'm going in these people more collaboration.
Yes, the more we are, the better for the world and for the benefits for our health.
Okay, if you like to collaboration with the Tropical Disease Center from Indonesia, maybe I want to send to you about the paper about collaboration with Dr. Stephen and so on, the team, the HMUH convention next. Maybe I want to send to you about the result competition result HMUH. Yeah, thanks. About this is I like it if you collaboration the Christine. Thank you, Dr.
Thank you.
Stephen. I like it.
Thank you.
Okay.
Thank
Okay.
you. Thank you very much, Professor.
Thank you.
Thank you. Okay, Cristina. Now we have 20 more minutes for your next presentation and discussions.
Okay.
Okay.
Ooh, again, the same. Okay, just wait for me a minute. Okay, I will. Didn't I stop the sharing?
No, you stop the sharing.
Yeah, okay, but it's I will try the new one. Yes, I will do. I have some problems with my yes, just a moment. Yeah, I will try now.
No problem, no problem, no rush.
No, I don't want to rush, but I want to be in time.
Yeah, now it's coming. Yes, and
there is something with my mouse. Something is not good with my mouse. So we know that we are all concerned about how we can find a better natural way to protect ourselves or to treat in case we got the so bad fame COVID-19 disease. So we know bee products have been proved to be successful in such treatment, but unfortunately, not all of them have been carried out in clinical trials, which are the only one which are recognized. I want to point out that this presentation is just the result of the work of the team of Dr. Ionuț Moraru, whom you have already introduced into your message on the WhatsApp, and also of his wife. And I have contributed together for making this presentation related to several other natural compounds or products that can be used together with bee products. So this is a stage of what is happening with bee products and scientific evidence on the viral anti-activity of them, especially with focus on SARS-CoV-2. The ongoing clinical trials against SARS-CoV-2 are only valid with honey and propolis. And we have a study on propolis that have been carried out in Brazil on patients that were sick with COVID, and also honey, but honey not alone, only together with another vegetal material like Nigella sativa. The other studies have been the preclinical, in vitro or in vivo, that on the antiviral activity, but do not include SARS-CoV. Then there are clinical evidence for, you see, honey, propolis, royal jelly, and pollen, but for prophylaxis, excluding also SARS-CoV, and the clinical evidence, unfortunately, it's only made with antiviral without SARS for honey, propolis, and only honey and propolis. Okay. What are the antiviral compounds? Probably this will be repeated several times that are present in bee products. And what are the main mechanisms of action of these compounds? The inhibition of viral proteases, which are the enzymes that make the virus active, are the phenolic acid, the flavonoids, and the polyphenolic ester, caffeic acid phenethylester, the so-called CAPE, whatever you want to call it in English. The virus inactivation is produced especially in honey by hydrogen peroxide, which is produced during the glucose oxidation in honey. And we know that so this hydrogen peroxide has been used as an antibiotic, but also the so-called inhibin in honey. That was long ago when honey has began to be studied as a potential pharmaceutical product. The activation of antiviral immune responses, especially in honey, have been shown that levan, a beta-2,6-fructan, which is a polysaccharide produced by fermentation with Bacillus subtilis, and the ascorbic acid can produce this activation of the immune responses. The rosmarinic acid that has been identified in several types of honey can binds the S-RBD and blocks the interaction with angiotensin converting enzyme, the gateway for the virus to come into our body. To reduce the viral load, there are flavonoids like rutin, quercetin, and isoquercetin, in which are abundant in propolis. You will see in the following presentation probably the level of quercetin in propolis, but also in honey and bee pollen. Mechanisms that have not yet been defined are some related to the fatty acids in honey, the 3-hydroxysebacic acid. Also, we know that honey is credited with no fats, no lipids in it. The phenolic acid paracumaric acid and benzoic acid, we know it is also an antibacterial and antifungal agent. This is a small scheme in which we try to show how propolis acts on the host and the pathogens. It's somehow recreated from a published study that shows how propolis acts. So on the host, the propolis works on immune modulation by stimulation the innate immunity and also modulation of the pro-inflammatory responses. But also propolis can maintain the status of the cellular antioxidant system. So it is important from both immunomodulations and the antioxidant activity. At the level of the pathogens, the propolis pathogen penetrate into the host cells, but the propolis used inhibits the key enzymes and the proteins responsible for the penetrations into the host cells. In respect of the replication, one in our body, propolis intervene by inhibiting the key enzymes responsible for the replication of pathogenic genetic material. And in respect of the metabolism of the pathogens, propolis inhibits the energy-producing enzymes at the level of the mitochondria and produce what? The disruption of the cell wall of the pathogen. Propolis and honey flavonoids, example, rutin, naringin, cape, luteolin, and artepilin C in green Brazilian propolis, can inhibit viral spike protein fusion in host cells, virus-host interactions that trigger cytokine storms and viral replication. Similar to remdesivir, a routine ethanol propolis extract and propolis liposomes, about I have already spoken, can inhibit SARS-CoV-2 non-structural proteins in vitro. That was the results of the study. And these compounds, together with naringin, inhibit SARS-CoV-2 infections in viral E6 cells that have been used in the study. So, as the propolis extracts provided by nanoparticles have better antiviral effects than the ethanol extracts, we should try these pathways, probably can offer better results. Similarly, in the study with honey and the Brazilian propolis and the combination with Nigella sativa reported an earlier viral elimination, recovery of the symptoms, discharge from hospital earlier and lower mortality than their counterparts who received only the standard care into the hospital. Now I will come back to the bee product, possible honey and royal jelly, but 10-hydroxy-DCA decinoic acid exists also in these compounds, can make an induction of leukocyte attachment to viruses with their destruction. And also, this compound can promote the maturation of dendritic cells derived from human monocytes and the polarizing ability of T helper type 1 T cells, strengthening the antiviral immunity. So we have here the picture of the bee products. But there are several natural products that can be used together in synergy with bee products to offer better results. I'm speaking now about well-known natural probiotic Kefir, which can be used in several formulations together with bee products. They show antiviral and immunomodulatory and anti-inflammatory activities, and the active compounds involved are the polysaccharides, proteins, and peptides, macro elements. We know Kefir is very rich in such both macro and micro elements, essential amino acids, the vitamins B1, B2, B5, and C, fats, and also the secondary metabolites like catechin, vanillic, ferulic, and salicylic acids, and also a very, very important product, which is kefiran, specific to kefir, microflora produced by Lactobacillus kefiranofasciens. What are the mechanisms? First, the inhibition of angiotensin converting enzyme, inhibits the viral adhesion, suppresses viral activity. From immune point of view, this kefir can increase the production of G lymphocytes and immunoglobulins, improving the expression of cytokines, interleukin-1 alpha, IFN gamma, tumor necrosis factor alpha, interleukin-6, and interleukin-10, which is specially attacked by kefiran. From anti-inflammatory activity, it's the inhibition of the cytokine activity, such interleukin-1 beta, tumor necrosis factor, and interleukin-6. Colostrum, we all know these very, very precious products that we got from our mothers with the first milk that we suck. So colostrum is the main source of immunoglobulins, and with it, we start our life with the best nutrition. Colostrum shows antiviral, immunomodulator, and anti-inflammatory activity, and the main active compounds are lactoferrin and lactoperoxidase, as well as lipids from which anti-infective degradation products are generated during digestion, and antimicrobial peptides. There are very small peptides, very active, but very important are the immune globulins, IgG, the gamma globulins. Some combinations or ingredients and their biological action mechanism with their active compounds can also have good results in association with the bee products. I will introduce the main compounds in a well-known product in Romania that had been developed by Dr. Moraru, and which really has been used by a lot of our colleagues in Romania that are practicing apitherapy. And this is an important combination of 9 mushrooms traditionally used in Japanese medicines that influence the immune response, the natural killer cells, the macrophages, the T4 helper, and T8 delayed hypersensitivity lymphocytes. They have a prolonged effect on cellular immunity and stops the cell invasion and viral replication that has been seen in the treatments carried out by Dr. Moraru, that the effect on cellular immunity is prolonged even 10 days after the treatment with this product has been ceased. The active compounds are the polysaccharides, complexes of polysaccharides with proteins and peptides, enzymes like ribonucleases and proteases, and lectins. There are also the secondary metabolites already mentioned, like low molecular weight compounds, lactones, terpenoids, and alkaloids, not the bad ones, antibiotics, and metals that act like chelating agents. Then a herbal support for viral infections produced by spagyric technique containing stinging nettle, astragalus root, echinacea, the aerial parts, and the root of licorice, shows antiviral, antibacterial, antioxidant, immunomodulatory effects, and inhibition of viral replication, especially of licorice root. And these are due especially for astragalus for the polysaccharides with immunomodulatory and antioxidant properties. Another proposed ingredient or combination is that of fermented plants like black radish, Raphanus, and licorice, plantain, Grindelia robusta, Daucus carota, and black pepper fruits. They are also used in this combination of the product. Another very important, and that has been mentioned in several presentations and intervention yesterday, is the essential oil from oregano, which has strong antibacterial, antiviral, antifungal activities, strengthen the immune system, which proved to be very effective in COVID-19. And the main compounds that have been studied, and we know many, many details about them, are carvacrol and thymol. The black cumin essential oil, Nigella sativa, is also immune stimulant, antioxidant, anti-inflammatory, but has also some other important activities like the antitussive, bronchodilators, and antihistaminic agent. We have already mentioned the clinical studies that have been carried out with honey and Nigella sativa. Here are the omega-6 and 9 fatty acids that are found into the seeds of Nigella, vitamins of the A, B, C, amino acids, enzymes, and minerals, especially not only the macro but trace elements, saponins and nigellones, which are specific substances from which the most study was thymoquinone. Another ingredient, natural ingredient, is the honeysuckle extract, Lonicera japonica, which prevents influenza A virus infections with all the variants like H1N1, H5N1, H797, but also against SARS-CoV-2 and herpes simplex. These are credited with the existence of some uncoded mRNA nucleotides and the microRNA 2009-11 from the microRNA classes. The horseradish, Armoracia rusticana, which unfortunately in the EU Register of Health Questions of Health Claims has no such claim permitted. It's the antiviral, antioxidant, and the regulation of the inflammatory function and the direct antimicrobial properties. The main compounds also are glucosinolates, but also the microRNA, the same, the code 2911. Shilajit, or mumio, it's the so-called mountain resin in Himalaya, which is very important because of its very rich compounds in minerals and not only resins, which supports the normal functioning of the immune system. Silver and the essential oils like eucalyptus and oregano, together with propolis tinctures, can offer very good antiviral, antiseptic, and antifungal effects. Of course, we know silver has always been an antimicrobial, a strong antimicrobial. That's why the kings were using their cutlery in silver. And also the active components of essential oils already mentioned for oregano. A very interesting product with highly nutritional action and a wonderful immunomodulator activity is the product called Amrita, which is a kombucha fermented polyflower pollen with consortium of bacteria and yeasts. Amrita, the so-called food of God, was also obtained through a research international project by Dr. Moraru and his team. The high content in probiotics, polyphenols, flavonoids, silicon, proteins, and essential amino acids make out of it a very rich and important ingredient in products like biotics with different destinations, target organs, but especially very useful in COVID treatment. Another product, which is a result of an international research product, which is also on the way now, is a combination between colostrum and kefir. Actually, it is fermentation of colostrum with kefir granules, which offers a wonderful modulation of the immune systems, helps maintain the normal intestinal flora, which is so important in fighting COVID disease, but has also important gastrointestinal and cardiovascular effects. This called kefir, already registered mark, is a prebiotic, meaning that it shows both pre, po, and postbiotic activity. It has extremely high bioactive peptides like lactoferrin and immunoglobulins, transfer and growth factor. Of course, we should not neglect the vitamin C and D, which support immunity. I will not mention here the claims written in the Regulation 432 on 2012 in Europe for the accepted health claims for food and food supplements. That is all. It's not all, because we have always a lot of other things to discover, and in our experience, those as medical doctors or other healthcare practitioners can find more and more natural products that can be used together with the products to increase our immunity and to help fight in case of disease. Thank you very much for your attention.
Yes, thank you very, very much also, Cristina. Yes, you can stop now the share screen. Very good, very good. Now, yes, another very, very good presentation. So it was wonderful to see that there are so many natural products which can have direct antiviral effect and also good for the immune system. So it's two ways. And to combine these products in one product like Dr. Moraru is doing, to have a very good synergy is wonderful. Okay, questions from our participants. If you have questions, please raise the hand, put the yellow hand up. You have it here in the reactions part. And you can put any questions you have. If not, let's go to the next presentation. I believe it's also with us, it's Otilia. And maybe we'll have the free discussions in the evening. We'll have some more discussions.
I think the Dr. Moraru, who will also join probably the free discussions, will offer more and more details about his own study on this and the results that he got. I think it's very important. Anyway, I thank him for giving me the possibility to present these results. Okay. Thank you again.
Okay, thank you also, Cristina. And now let's give the floor to Otilia, who has presented also a synthesis on some of the best biopharmacological properties and composition of the B products. Otilia, you can activate your microphone. Okay.
Yes. Thank you, Stefan. Good morning to everyone. I will try to share. Please give me the permission. Okay, but why I cannot share? Host disable participant screen sharing.
Oh, yeah, you went out. Yes, sorry, you went out for a while. So now I put you again co-host. Sorry, Otilia. Okay. And the system deactivated. Now we can share.
Happy conference.
Yes, it's coming.
Good.
Perfect.
Yes.
Okay.
Hello, dear friends, again. My name is Otilia Bobis. For those who do not know me, I am a senior researcher at the University of Agriculture Science and Veterinary Medicine from Cluj-Napoca, Romania, and also working in a laboratory for quality control of bee products. Let me give me the opportunity in the next minutes to give you a short overview upon bee products, which are, as we all know, important sources of bioactive compounds, and some topics about the bioactive compounds that we have here in bee products, the quality properties, and of course what is very important, problems, short problems of labeling. Okay, we all know that the main bee products are honey, pollen, bee bread, wax, royal jelly, and propolis. The question is which of these products have the most important amounts of biological active products, because now we are talking not of bee products like a nutritional point of view, but also for their properties, for their biological properties. So what are the biological properties in our bee products, and what type of substances are there? Okay, sorry, I did something.
No, it's okay. It's okay now.
Okay.
Continue. With the mouse, you can rotate the mouse for the next slide.
The mouse, yes. So it is known that nutritional substances in all food products, in bee products as well, are sugars, lipids, proteins, and the amount of water. With these components, we can calculate the nutritional value of the food and also of the bee products. But biologically active substances are different classes of substances which are found in minor amounts, in very small amounts, and these classes may be organic acid and vitamins or enzymes, phytohormones, natural pigments, and the most important, I would say, are the polyphenols, but also not the last, essential oils and different aroma substances. When we talk about each of bee products, let's start with honey. I say that polyphenols are the main responsible substances for the biological activity that we all know. Using different motor techniques like high-performance liquid chromatography, we were able to detect and to quantify in honey different polyphenolic type, like phenolic acids, as you can see, caffeic acid, paracumaric acid, ferulic acid, parahydroxybenzoic acid, and different flavonoids as aglycons like apigenin, like camphorol, like crisine, sometimes quercetin. What you can see here are two chromatograms from two types of honey. So different substances can be found in different types of honey, from which derive the different biological activity of certain type of honey. Going further, from propolis, using the same HPLC methods, we also quantified a lot of biologically active compounds from the class of polyphenols. We can also see here caffeic, paracumaric, ferulic acids, and also higher amounts of flavonoid like quercetin, like camphorol, pinosembrin, crisine, galangin, which Cristina was mentioning earlier, which have very good and very high bioactive properties. Also, an important component of propolis is caffeic acid phenethylester, shortly CAPE, which have very high anti-cancer activities. As you can see here, high amount of quercetin we have in propolis. I mention only the properties of only these two types of flavonoids, quercetin and CAPE, with very, very good biological activity, and especially they are very much let's say analyzed these days when this pandemic situation is going on. Unfortunately, we have more than 2 years with it. So talking about quercetin, we all know quercetin as a food supplement presented in many ways, in pills or in capsules, or extracted from natural sources. This should be like that, so not chemically synthesized. So quercetin is an important flavonoid which is found in plants, in all plants. It is found, but also we have detected it in honey, and as you could see in propolis in higher amounts. We can use these supplements, of course, but we can use our bee products as important sources of quercetin, and not only because sorry, we will have this synergic effect if we use bee products instead of this single substance supplements. Why quercetin is so used as food supplement? Because it have a lot of biologically active activities like antihypertensive, anti-inflammatory, anti-atherosclerotic, anticholesterolemic effects. And not only this, because studies were made, and lately in the last 2 years, a lot of studies were made, and quercetin was demonstrated to have also a very good antiviral activities. If we go further with our bee products and the biologically compounds that they contain, we can see that pollen is a very rich source of vitamin C, fresh pollen, so crude pollen or fresh, like we say, but also a very good source of carotenoids. Lutein is the most important carotenoid in pollen, but also beta-carotene and beta-cryptoxanthin are present, and they have a lot of important, wonderful, let's say, activities. Pollen is product which contain more complex polyphenolic substances, so we will not find pinosembrin or crisine as ugly cones, but linked two different pieces of sugars, and you can have here and read what derivative, complex polyphenolic derivatives can contain bee pollen, mainly fresh pollen. We are going further. As Cristina said also earlier, royal jelly is known especially from the composition, the presence of 10 HTA, so hydroxydecenoic acid, very important fatty acid present in high amounts in fresh royal jelly, but also for these major royal jelly proteins, which are indicators of authenticity and freshness for royal jelly. Okay, now all these compounds we have to be able to quantify. So the quality of bee products is made with lab analysis, and a full spectrum of analysis is needed in order to have an overview about the chemical composition and also about the properties of each bee product. So regarding honey analysis, it's very important to have also physicochemical analysis, so for macro components of honey, and also different types of analysis for bioactive compounds. So water content, ash content, electrical conductivity of or acidity are important. The diastasic index, which is a freshness component giving us the amylase content of honey, also peroxidase is very important. HMF content, which give us the view about the freshness of honey, or if it's not adulterated or do not have like I don't know, storage, wrong storage methods. Sugar spectrum also is very important, and as I tell you, using some modern techniques to be able to quantify the bioactive compounds from different classes, which I have mentioned earlier. Also very important, because yesterday also, and I believe today also will be talk more about unifloral honeys. Melissopalinology is a method, a specific method for botanical origin determination, in which we establish if pollen from the specific plant which give the name of the honey is present in the amount that are specified by national or even international regulation. We have here in Romania some types of honey, including acacia, I think sunflower, roseberry, and lime, which are stated how much of pollen grains should be in these monofloral honeys. We are working together with Cristina, with Mrs. Cristina Mateescu, and with Alina Parabi and other colleagues from my university in the group of experts from ASRO Romania to develop a draft for bee products, international standard for bee products. And I hope that for botanical origin determination will be fruitful discussions in the future for having this. For propolis analysis, water content, ash content, bioactive compounds like total polyphenols, flavonoids, and individual polyphenols, and also the antioxidant activity are stated in the draft, in the international draft of propolis standard, which we have to follow if you want to commercialize or to have to even to consume these bee products. And these analyses are specified in the propolis draft. For royal jelly and wax analysis, also physicochemical analysis and the bioactive compounds classes, major royal jelly proteins and 10-hydroxydecenoic acid. And for wax, of course, the falsification is very important, and also the presence of contaminants, because as we all know, wax is one of the most let's say absorbable, I don't know how to express in other way, bee product with contaminants. So in wax we found the higher amount of contaminants, even we talk about antibiotics or we talk about insecticide, pesticide, acaricide. So wax is the bee product which retain more this type of contaminants. Okay, the laboratory in which I'm working is accredited for quality analysis, and we are registered in a proficiency scheme with other laboratories from all over Europe. Also, we can perform in the lab, besides the physicochemical analysis, bioactive compounds from bee products and other natural matrices, we can perform antibiotic analysis and heavy metals determination. Okay, now we reach bee product labeling. Labeling of bee products is very important. It's a very important issue. And I want to mention that last year, I think, or in 2019, our research group published a review regarding labeling, regulation, and quality control of honey. It was a review study published in a very important international journal, Food Reviews International, as for the other bee products, as I told you, for honey also there is a group of experts which work on the developing of an international standard, which I believe is very helpful for the future, for all of us, even if we are beekeepers or just consumers or traders for bee products, to have the same, let's say, analysis methods, and also to have the same quality for bee products that we import or export. So I will talk a little bit about labeling of bee products in Romania. Because labeling should follow the legislation in each country before we will have this international standard. Due to the fact that honey must have on the label, as every food product, the nutritional value, so the values which give the nutritional and energetic values, there are a lot of preprint, let's say, labels, which you can find it on internet. But what is the most odd thing is we have here linden honey, which have 340 kilocalories, and it have 30 grams of sugars in 100 grams of honey. Wow, that's odd. We have also a label of multifloral honey, which have, yes, 340 kilocalories and 30 grams of sugars. 30 grams of sugars with 20 of water are 50, and what are the rest 50s? So I'm advising the beekeepers, producers of honey to take a little amount of money to go to the laboratories which can make this analysis and to make labels of their own. So it's very important to have this mark, your mark, because you are a producer, you are a producer who make these wonderful products, and it's a pity to have these stupid things I'm saying. I hope I'm not too hard on this, written on the product that you produce and you want to sell. It's not a huge amount of money, and you will have your own brand, and you will be in line with the law that said that every jar of honey must have the nutritional value, and you will have the nutritional values for your own producing honey. If we go further, pollen. For pollen, we do not have such wonderful things written on the label like for honey. Maybe because pollen is a more complex bee product, and not everybody can guess, like for honey, how much amount of proteins, how much lipids or sugars are in there. But for the pollen, of course, it can be made also this nutritional value etiquette, which will be put on the label. But I think that the most important problem that it should be written on pollen, if we are talking about dry pollen, or if we are talking about fresh pollen, it's the content of water, because as you know, pollen may be very easily, if it's not dried properly, or if it's not kept in the freezer, if it's raw, if it's crude propolis, it can be attacked by very quickly by molds and yeasts, and we from this wonderful product, it can be transformed into something very bad. So I say that energetic value also can be written on the label of pollen after, of course, doing this analysis in a proper laboratory. Okay. So we reach propolis. Propolis is known and used especially as a tincture in different concentration, but mostly 30%. Everybody knows how to make this 30% of tincture, but my advice, I was listening to Cristina earlier. My advice from the experience that I have in the lab is to use not 96, but 70. And I believe that the ISO standards states that 80% of alcohol should be used for determinations in producing the tincture, even in the analyses that are made for raw propolis. And as Cristina said earlier, there are some flavonoids, that some polyphenols that are extracted in water, and for this reason even the new standard will state that 80% of alcohol should be used in the production of this tincture. What I am presenting you here is a propolis tincture, which is made from purified 95% of propolis. Purified propolis. What is this purified propolis? You can see it here. Purified propolis is made by boiling raw propolis in water for some, I believe, some hours to remove the wax. After that, this mixture is cooled and the wax is taken away. What is remaining is this dark, almost black resin, which smells very strange, which do not have the aroma compounds and volatile compounds from propolis, from raw propolis, and of course it has no wax. That's for this reason it said purify 95%. What should anyone do this? For commercial purposes, especially. Why? Because this purified propolis, if we put it in alcohol, in a few hours, it will be completely dissolved. And we will get a very dark solution, which do not smell like tincture of propolis, but it's obtained very quickly, and it can be diluted several times because it's very dark in color to reach the commercial color of propolis. Doesn't matter the composition, doesn't matter the biological activities, just commercial. We all know that if we make propolis tincture, we have to wait at least several weeks to get this solution. So it is very easy, but it is not good to use this kind of propolis, because in the process of boiling, we definitely lost every aroma and every volatile compounds that the resins, which the bees are working so hard to collect, will be gone, and we will not have a true, let's say, a true propolis tincture which can be used. And it was so much discussed in this situation, and also in other situation, other diseases which we can use this wonderful product. So in order to summarize, in order to have the best bioactivity of our bee products is to maintain a balance between the processing of bee products, and also it is important the colony treatment, because bees, like all other being, not insects or how we call them, they are getting sick and they must be treated. But the colony treatment must be made in such a way that we do not have residues in our bee products, and we limit the processing in the minimum level that we can. In this way, we will have good bee products, we will have happy bees and happy people, because they are using clean and very good products. Thank you very much for your attention.
Yes. Okay. Thank you very much, Dr. Otilia Bobis. Very good presentation. One short, small detail, practical detail. I saw that you mentioned that we need to measure in the bee products, honey, propolis, the pH. And
Yes,
yes, and this is important when we treat patients with COVID or people having pneumonia, because we need to have a very strong antibacterial effect. And we know that propolis, at least, propolis or honey, if you get from the acidic pH, you go to alkaline, the efficacy of honey and propolis against bacteria diminishes. So we should not put honey and propolis into milk, which is alkaline, when we want to treat a bacterial disease. What do you think?
yes, of course. We all know that honey is not indicated in people with gastrointestinal disorders, especially because this acidity, you know, it's very strange, because lemon is acidic, yes, but you can take lemons as much as you want, because in the stomach, with the stomach acidity, they will be neutralized. Honey, because of this acidic environment, it's not so indicated in high amounts. But pH and acidity, I was mentioning more or less because it's a method for authenticity testing. Because we all know that they are acidic, they should be in a let's say not so broad pH interval. And for this reason, in ISO standards, it is put this method. I don't know for medical purposes how it should act, but I know that this is a method for authenticity.
Okay. Yes, in case of people having gastritis, like a bit of disorders into the stomach or large intestine, small intestine, and so on, one of the methods we use in practice is to dilute the bee product in enough liquid. It's because honey, pollen, propolis, they are very concentrated, and when they come into the stomach, they absorb a lot of water. You know, honey is hygroscopic. Pollen is also hygroscopic.
Yes.
So there is a mistake which some beekeepers they do. They write on the labels, take pollen before meals or before breakfast. And this is a mistake, because usually before a breakfast or before a meal, we do not have so much liquids into the stomach. So the pollen will land on the stomach mucosa, honey too, absorbs the water and irritates the stomach mucosa. And this causes some pains, some discomfort. And some of them, they say, oh, I'm allergic to pollen, or I'm allergic to honey, or I'm intolerant, but it's just a mistake. You need to dilute it before, during, or after. And we have, maybe it will be also here in the afternoon, Professor Mamdouh Abdul Rahman. He told us that we can take even 150 grams of honey a day. If you take it with enough water, it's no problem. And I made also a test. I took 500 ml of honey in 1 hour, but with enough water. Okay, so I see here some questions. about the freeze drying of the substances. What do you think about this? Freeze drying would affect the quality of the product or not so much?
No. Freeze drying is a very good method for having this powder. I'm seeing that also Cristina responded in chat, but from all freeze drying means lyophilization. Yes, and to have this to remain with these properties, freeze drying is one of the best methods to preserve.
Okay, thank you very much. We have a question from Stephen. Stephen, can you hear us? Stephen, can you activate your microphone and maybe also your camera? We see that you put the yellow hand up. Stephen, are you with us? Or maybe he has some technical problems to connect.
Okay.
Yeah, and we have another one from Frederick. Okay, maybe Frederick will be faster. Frederick, yes. Please tell us your question and activate your microphone, Frederick. Yes.
Hello. Thank you very much to Dr. Otilia for the wonderful presentation you have given unto us. Now, my question is just on the laboratory testing of the bee products. Now, what are some of the general international parameters other than the contents of the bee products looked in during laboratory quality analysis? Thank you.
So as I said, there will be a list of quality control determination in the international standard, but also in the lab. And as I said, in our lab, different antibiotics must maybe determine. We have methods for this, and also heavy metals like lead and cadmium, which are the most dangerous in bee products, we can determine. And for bee products that are used with medicinal and pharmaceutical purposes, it is highly demanded that these contaminants should be checked. So beside the physicochemical and quality parameters, contaminants are very important, and also some microbiological determination. As I told you, especially for pollen and bee bread, some yeasts and molds count. It's important to be made.
Okay. I saw also a question, Otilia, in the chat area, if you can do now or in the future also analysis of bee venom.
In the future. because all the components that are present in bee venom, so in lyophilized bee venom, I guess, or even in raw bee venom, but this is harder to preserve. And bee venom, as far as I know, it is preserved as a lyophilized powder. So this could be subjected to I don't know, to be counterfeit, you know, and this authenticity determination should be made. But we have to look in the literature for methodologies to see if what equipment we will have in the lab are good for this, and to develop of course methods for this. But this is something for the future.
Yes, yes. And also I saw in your presentation, you mentioned that sometimes you can analyze some individual components in propolis,
yes,
like pinosebrin and so on.
Mm-hmm.
And this is very important for us as medical doctors, because we like to have this kind of thinking, like substance and effect, substance effect.
Yes,
So it helps us to the promotion.
Of course, the amount of each compound it's very important, and that's why I said different types of honey, or different even different types, or propolis samples are different in the quantity of bioactive compounds that we found.
Excellent, excellent, very, very good. Any other question or comment to Otilia or to Dr. Mateescu? She's also here with us. I see that she's giving answers here in the chat area. We can put also fresh pollen in the yogurt, fresh yogurt, kefir, 12 hours overnight. Okay, to improve the probiotics part. Yes, very, very good. Okay, thank you very much to all of you. Thank you, Dr. Bobis and Dr. Mateescu. We take the lunch break, and it will be a bit shorter. We come back at 2 p.m. Yes, it'll be 45 minutes. And please come back. We'll continue also with more wonderful presentations and wonderful speakers speaking about the protocols and so on and so on. Thank you very much again, and have a good appetite during your lunch or dinner or breakfast.
Okay, thank you. Bye. See you.
Okay, thank you. Bye-bye.
See you later.
See you later. At the same link. So you come to the same link. We already have the same link in Zoom. Will be also valid when you come back in 45 minutes from now. Okay.